So one thing that I always like to do, and like Dr. Holt, I'm also a big fan of cattle. They are amazing, amazing creatures. And I like to start right here, almost as much to remind myself is like people need to eat. It's one of the most essential things that people need to do. And cattle are an amazing way to feed people because they can do such amazing things with the resources that we put in front of them. I do not like eating grass. It's not a lot of fun, but I do like eating cattle. Making cattle production work better makes the lives of people better, even people who don't eat cattle. And I think it's important to remember that because they do all kinds of things for humankind that even if people don't eat them, they're still providing those environmental ecosystem services. And then part of that for me as a veterinarian and someone who's involved in cattle health research is I think if we can make cattle health better, we're going to make this system work better so that it works better for the producers, for the people who consume that food, all of those things. And I don't have any good dog pictures. My only real connection to cattle, I live on a little acreage and mostly I have sheep, don't hold that against me. But my uncle-in-law raises Simmental cattle, and that is the little Simmental calf that was born last fall. His name is Winston. So everything at our house gets a name, whether I want it to or not. So what I want to talk a little bit about today, I'm going to get into the physiology a little bit deeper, but I think one of the perspectives that everybody that's talking this morning is going to share is that what we would really like to do is figure out how to make animals healthier by figuring out what we can tie the associations between characteristics of the animals and the environments that they live in and the things that we do to manage them and the outcomes that we're interested in. So we're very interested in heart failure for this particular situation, but we've got a number of important problems when we try to make these linkages. There's a lot of things. The first and probably primary problem that we all focus on is that there's a lot of things that are associated that are not necessarily causally related. So rooster crows every morning, that doesn't make the sun come up. Sun coming up might make the rooster crow. That might be a little more causal. The other problem that we deal with a lot is that there's a tremendous amount of variation both in the animals that we're trying to figure out what characteristics are driving these diseases and in the diseases themselves. So we see a lot of variation in not just the manifestation of a particular disease, but often in the paths that can lead to the development of a particular syndrome, which heart failure is probably it's a good definition of a syndrome, something that is a process that there are a number of things that feed into it. So we're going to try to deal with animal variation and outcome variation while we're trying to make an association. And the goal of the research team that I'm part of is we really want to find causal relationships. We start with associations, are two things happening at the same time and we think they're pretty related to each other. But what we'd really like to find is a light switch or something like that we can turn off to turn off disease. That's one of the major goals of our research group. So we've already mentioned that cattle are cool. We all have one of these, and the cardiovascular system is a fascinating, fascinating system. If you like fire and flowing water, this is basically the flowing water part. I think that's a characteristic of every male in my family, both the fire and the flowing water part. This is such an amazing system that it sustains life. So what the cardiovascular system does is it is constantly delivering required nutrients to our bodies. At the same time, it is removing waste, toxins, products of metabolism, things like that. This thing is incredibly well orchestrated. I told Dr. Holt I've been trying to think of good analogies. This is like a relay race. And in order for this thing to work, the baton holder has to reach the person that they're passing off to at exactly the right time, and they got to be going the same speed in order to make that transfer. We've all seen relays, I've probably been part of them, where the holder isn't fast enough to catch up to the guy that's taken off. That's a bad deal, and that's essentially what's happening when there's not enough oxygen in the air. So the circulation is very well suited to adapt to that. But there's a couple of characteristics that you can think about when you think about how cardiovascular function works. First of all, the heart is it's not a very cool way to talk about it, but it's a muscular bag. It actually has no ability to generate suction as a pump. Many of you work with pumps in what you do. Those pumps can prime themselves, right? They can suck fluid in before they push it out. The heart can't do that. The heart is entirely dependent on that elastic vasculature. All those blood vessels stretch out a little bit, and once the heart relaxes, that stretched out blood vessel will push blood back into the heart. One of the ways we'll talk about in a minute when it fails is when that can't happen. The other thing is that that muscular bag can actively shove blood out when it contracts, but it can do it because there's check valves in the system. There's basically backstops that prevent the blood from going backwards while that muscular bag shrinks and shoves the blood out, hopefully downstream. Get a lot of pathology when that doesn't happen. Next thing that's important to remember, and Dr. Hold already mentioned this, is there's two circuits. We have one that's going to the lungs in order to reoxygenate blood, and then the other circuit is going out to the body and the organs, muscles, things like that to provide those nutrients and remove the waste. Every heartbeat has to have about the same amount of blood going both directions in order for it to stay in balance. So if you think about how much body there might be compared to how much lung there might be, there is a lot of blood that has to go through those lungs and through that right side of the circulation. Part of the way it does it is that the right side, when I talk about right side, that's the part going to the lungs, left side is going to the body. The right side circulation is supposed to operate at a lot lower pressure than the pressure that goes out to our body. So if you go to the doctor and get your blood pressure taken, they'll give you a number like 120/70. That's the pressure that's going out to your body. If you measured that in a normal situation, the average would be like 40 is something we would consider okay on the right side, right? Or below 40 would be better. So it's a muscular bag. There's two circuits, and there's check valves that keep that blood moving around in a circle. So there's both passive and active things that are going on at the same time. There's three big ways that this system can fail, particularly the heart can fail. One is the failure to fill. So if we block in this little, I'm not going to be very good at being able to show you what's going on, but basically those little black bars represent a blockade to filling. Interestingly enough, most of the time it's not a blocked vessel that prevents the heart from filling with blood appropriately. It's fluid or something that accumulates around the heart itself. I'll tease my next slides because I did this wrong. This is what I should have changed, Jackie. But examples of this would be heart failure or hardware disease. So when that wire goes through the diaphragm and pokes into that sac that's around the heart, what happens is it puts bacteria in there. That generates an immune response that brings pus into that pericardial space, and the sac that's around the heart is pretty tough. It's not near as flexible and stretchy as the heart is. So as fluid builds up in there, it pushes in on the heart, and it keeps that blood from being able to flow back in and fill it up. So blockade to fill can also happen from things like lymphoma. I don't know if you've ever seen a cardiac lymphoma. I found one of those in my career, and it's one of those things, when a vet says, ooh, that's cool, as a producer, you should be very concerned. All right, the next one is blocked outflow. This, I'm not going to spend any time on this other than to say it, because this is basically what Dr. Holt was talking about. When the lungs constrict, you can't get blood past it, and that will put a lot of pressure on the system and eventually make it fail. And then you can have just absolute pump failure. So we actually understand this pretty well too. Most producers have seen things like cardiac histophalosis. I don't know if you guys have ever seen that in your cattle, hopefully not. Or if you see rumensin intoxication, that will be damage to the muscle itself that results in the pump actually not being able to generate enough force to move that blood out. So those are the three big ways that the heart can fail, or the three generators of that. This is basically a summary of some of those causes. So I've gone through all those. High elevation and chronic BRD. If you get a chronic pneumonia case, those can cause that outflow blockage. Hardware lymphoma can cause the failure to fill. Ionophores like rumensin or lasalicid can cause that damage, and then some bacterial diseases like even blackleg can damage that pump itself. And then we've got this other one, this bovine congestive heart failure is what we call it, this thing we see in feedlot cattle where they, sometime between placement and finish, they do us a grand disservice and kick the bucket. So the thing that I have thought a lot about relative to heart failure and the physiology of disease is we often look at something like the heart of an animal and we get one look at it, right? If I can see the heart of a calf, he's not going to be using that anymore, right? Because it's out. We have tried to do like the echocardiography, like what they do on people to look at an ultrasound image of the heart. But it turns out that an ultrasound will penetrate about 4 or 5 inches. And it turns out that a bull in particular has a lot more than 4 or 5 inches of stuff to get through before I can look at his ticker. So that has not worked out very well. So the question is, what does the time course of disease look like? Another piece of the perspective is sometimes we look at a snapshot and we don't appreciate how the snapshot would have looked if I took it 3 weeks ago, or if I had the opportunity to wait 3 weeks and take it again 3 weeks later. So we do know some things about the progression of heart disease. There's quite a bit of work that's been done on this, and one of the most challenging things about heart disease is that in order to know when the insult happens, we have to figure out what the insult is, which is kind of that's the holy grail of research. So what we think happens is that there's an insult, and we have some evidence for this, but insult could be go up to 10,000 feet of elevation. The insult could be get a big old dose of Rumensin in a load of feed that maybe didn't get mixed very well, or develop a case of cardiac histophilosis, something like that. After that insult occurs, the heart has a tremendous capacity to compensate for that damage. That heart can, for instance, if the heart is surrounded by fluid that's preventing it from filling it appropriately, the heart will actually make up for that lost pumping capacity. Its stroke volume, the amount of blood that leaves every time the heart beats, is down, so it beats faster. So instead of beating 10 times in a certain period of time, it'll beat 20 if it's got to move the same amount of blood. So there's that compensation phase, and part of the compensation that we see in cattle is their ability to build muscle in order to overcome some of the problems that they're experiencing. So at elevation, cattle will actually build cardiac muscle mass to make the pump stronger so that they can push that blood past the obstruction in the lungs. And we see this compensation, and one of the things that our team wrestles with quite a bit, we talk a lot about, do we see different grades? So I've got a picture, and you'll probably see this, I'm certain you'll see this again, but the heart failure scores are out there. And when we look at these heart failure scores, we've asked ourselves the question, are we seeing a different timing situation where we've got an animal, particularly when it gets to harvest, it didn't die on its own? Are we looking at different stages of progression, or are we looking at different severities of disease? And I don't know that we have ever come up with a very good answer for that. The other thing is that I see pressure generated by the heart as something we can measure. And when we look at a case of heart failure, you'll see the red line in this graph. This is completely made up, by the way. I drew that line myself. That's not data-based. It's designed to represent what's going on and what has been measured. But as the heart fails, cardiac output, the amount of blood it can just move in total, goes down. And it starts going down more slowly, and that rate of decline gets faster, so they're moving less and less blood as disease progresses. But there's a period where we don't notice it that much because they're compensating. And that pressure curve that you see there is designed to represent that compensation. The heart is like, well, we got to build muscle, push harder in order to make sure that we can get that blood circulated around, make sure the relay handoffs are happening. But there comes a point when the animal decompensates and can no longer keep up, and that's when we start to see clinical disease in these animals. So we've got a timing problem, we've got variation problems, we've got the ability of an animal to essentially mask its disease for a very, very long time before we can see it, or I say very long time, we have no idea how long it's masking its disease, because in a situation like feedlot heart failure, we actually don't know when disease starts. All right, so case definition is one way that our group has really tried to deal with the variation around these diseases. So our group has picked terminal heart failure cases. So these are basically animals that the feed yard, the feed yards we've worked with are very experienced. They've watched a lot of these animals go through disease progression, and they'll say things like, that one's got 3 weeks, you can have him in 2. That was kind of the plan that we worked out with some of the feed yards we collected samples from. But we would choose animals that were terminal. And the reason we chose animals that were basically on death's doorstep is we needed fresh samples. We couldn't take them from a dead animal. That didn't work for us. But it removed the variation due to timing. So we didn't have to worry about whether the heart that we saw was at a different severity of disease, or if it was a different progression, a stage of progression at some point in that disease process, because we just took them all when they were just about ready to die. Then we also focused on the terminal heart failure cases because they're definitely the losers. They're the ones that are not going to make it to slaughter. And we didn't want to accidentally put selection pressure against animals that were successfully compensating for their disease. And then finally what we did is we opened all of these animals up and looked at their entire viscera to make sure we couldn't find some other reason that they had heart failure. If we had evidence of histophilosis or hardware or anything that we could detect, they weren't part of our study. So basically what it turns, so one other thing, in order to deal with the variation in the animals, the approach that our team used was basically these, I like this little example of the kid pictures. Find the differences. These pictures are almost the same. I didn't do this puzzle. That would have taken a while. I'm not very good at that. But basically our approach was to take animals that were very similar, same source, same sex, same color, and find the differences in their DNA code that were, the only difference between the animals was one was a case and one was not a heart failure case. And then we'd go DNA base by DNA base through their genome and look for patterns that were different. And our assumption was that those could be related to heart failure. And if we found a significant enough frequency of those patterns, we would say that might be a light switch that we can manipulate in order to change the outcomes for those animals. And we can get more into this later, but this is the payoff. We've got 3 pretty significant genetic risk factors. Though I say risk, the bottom one is a relatively new one. We just released that about a year ago. It was whole genome sequence discovered, so there are not even any SNPs on any chips anywhere. We're actually working with companies to develop the test for it. So I can talk more about this later. I think I'm nearly out of time. Thank you to my team. I say my team, this is the team that I am part of. Excellent group of people with a lot of skills. Especially thanks to Dr. Mike Keaton. We would definitely not be doing this without his genetics expertise. All right.